from Northwestern University, but he also did fellowships at Boston Children's Hospital with Mass General and Harvard. Chuck Bramlage is the CEO of Yamo Pharmaceuticals and is an experienced drug developer and has over 28 products that have been launched in 15 therapeutic areas to his name. It is a privilege to introduce both of these gentlemen, and I will leave it to you guys to dive in. Thank you.
Great. Thank you for the introduction. I'll start and just give a couple of slide introduction to Yamo. I joined in 2017, very interested. A senior scientist at AstraZeneca told me about this compound, L179, and he knows I like startup companies and asked me to talk to the inventor and the investors that had done one previous trial and were in the middle of a second trial. We completed that. Dr. Tom McGarrian, our chief medical officer, joined us at the end of 17. And we, of course, along with COVID and many other clinical operations people, were not able to do anything the last 2 years. We also had an issue with raising money. We're very excited to present the current data within the company, as well as what we plan to do right now as we start our third trial. We're very excited about it. We're interested in your feedback at the end of this. And of course, our expert Tom will walk you through the science behind it. We are seeking an additional $1 million right now of investment. And that investment would cover some more patients above and beyond the 50 that we're going for now to get 40 at the end. We think we can do another 20 to 25 patients with another $1 million. So those are the three things I wanted to leave you with at the beginning. The first slide here, we are a clinical stage company that's developing L179 to treat the core symptoms of ASD. We hold IP that goes to 2033 and other indications. We think this drug may work in post-traumatic stress disorder, type 2 diabetes, and fibromyalgia. It is a racemic mixture of alpha-methylparatyrosine, which decreases catecholamine synthesis. In 2018, we had a fast track designation from the FDA based on our previous 2 trials. We didn't have enough data to get the next designation, but we feel this current trial that we're starting right now is going to give us a lot more quality data from 5 top centers in the country with 5 experts in autism along with Tom. We're very excited to see what the results will provide. Next slide, please. I'm just hitting some of the highlights here. This month, we're starting this 50-patient double-blind study. We're targeting 40 completers in this. We're having a chronic dosing of 12-week with placebo and or a 2-period crossover with drug. These patients are 12 to 21 years of age with IQ of at least 70 and moderate impairments in socialization. These patients will be randomized between active, the 200 milligram BID, and 300 and placebo. We expect to get this information, the data completed by 2023 at the beginning of the year. We had just recently announced that Autism Impact Fund has become our first institutional investor. We've had all the rest of the investors have been high net worth individuals who have contributed and have an interest in doing something for these patients and their families. So we are seeking, again, another $2 million to cover some upside. $1 million will cover an additional 20 patients, and that's what we're working on now. Next slide. Okay, so here I would like Tom to describe the science behind L179, give you a summary of the first 2 trials very quickly, and more or less go quickly into the current trial that we're starting right now in 5 centers around the country. So Tom?
Thanks, Chuck. Can you guys hear me okay? My volume coming through, Chuck?
Yes.
Yep.
Yeah, thanks, guys. Appreciate the opportunity to talk to you about our exciting compound and what we think has some very good promise based on some preliminary data in an open label study and a phase 2A study. So alpha-methylparatyrosine is a competitive inhibitor of tyrosine hydroxylase, and what it does is to decrease the presynaptic release and stores of basically all the catecholaminergic neurotransmitters, which of course, as we know, are involved in many different functions, many of which overlap with some of the deficits seen in autism: memory, reward, attention focus, communication, pain, self-mutilation, repetitive behaviors. There's also a lot of indirect effects that the catecholaminergic system has, including co-release of growth factors, modulation of serotonin, acetylcholinergic, vasopressin, oxytocin, and other neurotransmitters. And we believe that this is a mechanism that works centrally as well as peripherally and might address some of the other medical and problematic concerns that are seen in autism, such as chronic constipation, sleep problems, abdominal distress. Next slide. This is just a kind of a mechanistic cartoon of where where alpha-methylparatyrosine hits. As you can see here in the drawing, it really hits at the early stages of conversion of tyrosine to dopa, which then goes on to impact the production of dopamine, norepinephrine, and epinephrine. Tyrosine hydroxylase, as you recall, is the rate limiting step. Is part of the feedback loop in terms of controlling the production of catecholamines. The reason that I think the inventor thought about using this compound was because of the fact that catecholamines are known to play a role of many neurotransmitters in regulating the dysregulated functions in ASD. In particular, many of us in the field know that dopamine receptor blockers, beta blockers, alpha-2 agonists do have some impact on improving some of the behaviors noted in autism. Sometimes those improvements do lead to improvements in socialization, but it's very variable. And we also know that we're not hitting the entire catecholaminergic, we're not modulating the whole catecholaminergic system when we use these drugs. We're kind of hitting various components. So the thought here is maybe maybe rather than hitting specific receptors, which are associated too with side effects, just down-regulating the overall tone of the catecholaminergic system might be a benefit. And that was the basis for this kind of broad spectrum approach. Next slide. So an open label study was done with 8 children. This was published data. Open label, all the caveats and disadvantages of open label being obvious. Sorry. The caveats of open label, limitations being obvious and mentioned earlier by our previous speaker. Nonetheless, we saw really a strong substantive majority, anywhere between 6 to 7 of the 8 patients who showed improvement in multiple assessment measures. In the ADOS, for instance, we saw anywhere from essentially 5 of the 8 patients showed a significant drop in their scores, some dropping to below the cutoff for autism on the ADOS. The clinical global impression assessment of severity also dropped by usually about 2 points, occasionally 1 point. And then the Conners parent rating scales also showed nice trends in the positive direction for multiple domains within the Conners forms. Next slide. So with this, the original team before we joined decided to do a 40 patient safety and dosing range study looking at 100 milligrams 3 times a day versus 200 milligrams 3 times a day in a double-blinded randomized fashion. Essentially what they showed was the drug was safe and well tolerated, which was the primary outcome measure, and a better response seen at the total daily dose of 600 milligrams or 200 milligrams TID compared to 100 milligrams TID. None of the efficacy measures were powered for statistical significance. And to be honest, only 10 patients received active treatment in each of the arms. So extremely small numbers. We were not, again, looking for statistically significant, but just kind of quantitative trends in the right direction. And in fact, we did see multiple measures, including the Vineland Adaptive Behavior Scales in the socialization domain, the ADOS-2, the CGI, the repetitive behavior scales, and the SRS showed greater trends in the treated over the non-treated for benefit in the patients treated for only 28 days. So this was a 28-day study, thereby demonstrating non-statistical but nonetheless clinically significant differences. Based on this information, the FDA granted us fast track, as Chuck mentioned. Next slide. This is the overall flow diagram of the study. You can see this was because it was a safety study and there was not a lot of, there was no preclinical data available, but because the L enantiomer of this drug had been approved previously, the FDA allowed us to go in with the racemic mixture as long as we did 5 patients open label in each of the dosing cohorts first, check for EKGs and labs, make sure there were no adverse events, and then allowed us to randomize 15 patients initially in the 300 milligram daily dose, 100 milligrams TID, and then in the 200 milligrams TID, 600 milligram total daily dose. And so we randomized after the safety cohort was completed, 15 patients, 2-to-1 active to placebo, first in the 100 milligram cohort, and then in the 200 milligram cohort. In the active group in the second cohort, the 200 milligram TID cohort, we had 1 child withdraw due to a single seizure. That was not an SAE because it was just an out of hospital seizure. And then a few people, 2 people, 1 person withdrew in the placebo group and the high dose group just because they had a parent decided not to continue with the study. Next slide. And so what we saw here was on the left-hand panel, this was a change in clinical global of impression in the 2 groups. The placebo group showed a 0.3 point change, categorical change, and the absolute change in the treated group was almost a full point, 0.8, so about a 0.5 point difference. In the lower dose, we saw a lower change, a half a point in the treated group versus 0.2. But again, a dose response, a higher degree of change in the CGI severity in the high dose compared to the low dose. Similarly, in the ADOS, there was a mean full point change in the treated group compared to really no change in the placebo group at the high dose. And in the bottom right quarter panel, you can see a half a point change in the low dose compared to, again, no change in the placebo. Not statistically significant because of the small numbers and variability, but still an interesting dose response change that was observed. And again, some of these changes actually were seen across multiple measures, as you see in the next slide. We saw similar changes in the Vineland Adaptive Behavior Scales in the socialization domain. You can see a 6-point improvement, go back up, 6-point improvement in the Vineland socialization for the first 4 weeks, with then a return to baseline, gradual return to baseline over 4 weeks over this ensuing 4-week period. And you see really virtually very minimal change in the placebo group. We also noted, did a kind of a responder analysis, and you can see that some of the response was driven in the high dose group by quite large changes in those who were treated, anywhere from 20 to 29 points amongst 2 out of the 7 patients that had a record. And then about 3-point changes in another individual, 3 points being kind of the minimum important clinical difference that we'd like to see with the Vineland. Next slide. On the SRS, we saw significant categorical drops from mild, in some cases from severe to moderate, from moderate to mild, and from moderate and severe to normal in those who were treated with the high dose. And the low dose, we really didn't see this level of change, but in the high dose, you can see on the motivation score, a 10-point drop compared to about 4 points on the placebo. Less difference, but still higher trends of dropping on the social communication T-score and the communication interaction T-score. Next slide. When we looked at the ABCs and the SRSs for particular areas where we saw responders being higher versus in the active group versus the placebo groups, you can see on the social withdrawal lethargy and on the inappropriate speech, we had patients responding on the active group about 40% and 50% respectively versus only 0 or 25% on the placebo patients. On the SRS awareness and social motivation, about 50% of the patients responded positively with a 2 or more point improvement. In social awareness, 60% for social motivation versus 20% and 0% respectively for the placebo patients. So when we did some sub-analyses, we saw much larger patients, much larger rates, between 40 and 60% response rates. Next slide. So we're actually just announced or in the beginning phases of initiating this study, which is a phase 2b crossover trial. We're starting off with 50 patients to randomize 40, or to complete 40, I should say, 50 patients randomized to complete 40. This will be total daily dosing of 400 to 600 milligrams, and it'll be BID dosing instead of TID dosing. And that's because we did some PK work in the interim between the last study and this study, and we're able to show a marked prolonged half-life compared to the L enantiomer. So the L enantiomer of alpha-methylparathyroxine has a half-life of about 3 hours. When you use a racemic mixture, you extend that half-life to about 12 hours, making it doable to dose twice a day. And so this will be an AB-BA study where half the patients will receive drug first and then wash out and go to cross over to placebo, and the other half will receive placebo first, wash out, and then go to treatment. And that treated group will be split between 200 milligrams BID and 300 milligrams BID, 50-50. We're also stratifying, kind of a soft stratification around anxiety, because we do believe anxiety levels might be a modulator of responses. And our primary endpoint is the Vineland socialization domain, looking at actually the growth scale values as the primary measure, and then the standard scores. We'll also be looking at some key secondary measures, including the BOSC, the CGI weighted for socialization, and then also the PGI, which is a parent caregiver endpoint where the parents will choose the top 3 symptoms of concern and then rate those on the same 7-point Likert scale as the CGI. We'll also be using the social responsiveness scale, social responsiveness scales in the various domains within, and then the PRAS, the Parent Rated Anxiety Scale, and looking at the Aberrant Behavior Checklist subdomains, as well as the caregiver strain and some of the other domains, all the other domains actually within the Vineland, so the communication domain and the daily living, activities of daily living domains. Next slide. So this is just a kind of a listing of the outcome measures that we're using. I think these are pretty well known by most of the folks in this group, but key secondary measures being the BOSC. We're excited to be able to use the BOSC because it's starting to be an interesting endpoint to be used that gives some of that structured play activity scoring based off of some of the ADOS modules and methodologies that gives a sense of outcome that is measured with a third-party involvement, utilizing third-party scoring. And we're excited about that because, again, many of us have used the ADOS to look for changes, but of course it's not a tool that is very sensitive or meant to be used for long-term outcomes and longitudinal changes, whereas the BOSC is. So we're very excited to be using that. And then also switching to a GSV, growth scale value responder analysis, versus standard scores. Many people know standard scores can still decline in a child with autism, but it's really that rate of decline and whether or not you can stabilize and have new skills learned on the Vineland that is important. So even if a child, for instance, is flat on a socialization T-score, but grows or expands their abilities more than the typical child with autism, that is something to be really, I think, taken as a positive outcome and should be looked at. So the GSV allows us to do that. Next slide, Eugene. So we have 5 sites that have been chosen and qualified and are in various stages of starting. We have the Thompson Center in Missouri, the Thompson Center in Southern California, which I am actually a part of. The PI there is Dr. Gola. We have the Center for Autism, SARC, in Southwest Autism Research Center, the Center for Autism in at Rush in Chicago, and then the Wheel and Columbia CAD-B Center in New York with Jeremy Beenstra Vanderwall. So very excited to be running this, and we have 3 of the sites initiated already and waiting for the first patient enrolled, hopefully by this month. And I'll turn it over to Chuck.
Yeah, so the long-term plan for L179 is, as we mentioned, to get the results of the data in early 2023. We would expect a 3-year development program before we would get approval for the drug sometime in 2026, early 27. So that's our current target that we have. We, of course, will go to the FDA, and we're hoping the fast track designation can help speed up the process. We obviously will do a compassionate use program after this with good results for those patients that were in the trial. We will also look to do other work that the FDA has asked us to do, which we can get into at another point. The last slide just points out, Eugene, if you could hit the last slide there, the team that's been put together the last 5 years really to do this work that we have right now. You've met Tom, Dr. Tracy Fisher came to us with lots of experience in clinical development and regulatory affairs. She's leading the reg function for us. And Wynn is the senior director of our operations and is running our trial, has a wealth of background. Tom used to work with her in previous roles. And Eugene is on the line now, our CFO. So we've got a small team. We would expect with positive results, we would grow the team another 25, 30 employees to help ramp up for the phase 3. So in essence, that's what we have. We'd like to turn it over for questions you may have that Tom or I can answer. Thank you very much.
Thanks, guys. Looks like Tom's addressing one of the questions that's come through via the chat, so thank you. One question I have you guys looked to see, given the dopamine theory of ADHD and what you guys are doing, have you guys seen, it doesn't look like there was much presented on changes in attention as a response to the intervention. Have you guys looked at that in any detail? Have you looked at subgroups of kids with autism with comorbid ADHD? I'd love to hear your thoughts there.
Tom, go ahead.
Sorry, I was muted. We did, in the previous study, look at the Connors, and in particular to see if there was any increases in hyperactivity. There really was no signal because we were worried about that as well. Is that, are we going to affect attention and focus? And did not see any worsening of that, across the 2 groups, but small numbers again. So we will be keeping track of that, especially with the ABC. So we'll have hyperactivity as a measure based on the Variant Behavior Checklist.
Okay. All right. Well, I believe that's it in terms of the questions. Pramila, is it time for a break? Thank you, guys.
We will be taking a break till, I believe, till 3 o'clock. And then start with one more presentation in the industry talks and then move on to more basic science.
All right. Thank you.
Thank you, guys.
Everybody.
Thank you so much.